Bismillahirrahmanirrahim. This is just a short video for your final revision before the exam tomorrow morning. First of all, of course, start the prayers with your prayers. Start your paper. Recite Surah Fatiha 3 times. Pray sincerely for guidance during this exam and every exam in life. You know the paper 2 tomorrow is for 1 hour and 45 minutes. It's an 80 marks. There is no section B and C which used to be in the old syllabus. Since June 23, the new syllabus, they are just short answer and structured questions. And there's no choice in it. You have to do all the questions. Quick recap of all what I want you to quickly revise. The 5 and 6 of this describe the structure of a bacterial cell, limited to ribosomes, circular DNA, and plasmid, cytoplasm, cell membrane, and cell wall, which is not cellulose, it is peptidoglycan, and the functions of the above structures in animal, plant, and bacterial cells. So you can get a comparison between animal, plant, and bacterial cells. Then I want to see the image and the magnification. You must always know that. In chapter 2, you must know the binomial system. I marked that 3 and 4, construct a dichotomous key. If anything is not clear to you, just Google that and go through the videos. If you have any videos that you can look into it. Then I want you to look at the structure of the virus, limited to protein coat and DNA or RNA strand, which is the genetic material. Understand that viruses only replicate in living cells. Then the important part is in the chapter on diffusion, movement of cells. Understand that energy for diffusion and osmosis comes from the kinetic energy of random movement of molecules and ions. Then investigate the factors that influence diffusion: surface area, temperature, concentration gradient, and distance. These are the points that you need to remember in a question which might come on this. Then these are, of course, I want you to look at these for paper 4. Then biological molecules, I want you to look at the chemical elements, and the main is the DNA, which is something new. It has carbon, hydrogen, oxygen, nitrogen, and it has a phosphate. Then the tests, and then the fact that I'm talking about the large molecules. You must know starch, cellulose, glycogen made from glucose, protein from amino acids, and lipids from fatty acids and glycerol. Then you must know this, the DNA is made from nucleotides. Then in the enzyme chapter, I want you to know they explain the effects of changes in temperature and pH on enzyme activity in terms of kinetic energy. Increasing temperature increases the kinetic energy, then the shape and the fit. The active site is complementary to the substrate, and substrate fits into the active site, and the fact that they're denatured at temperatures above the optimum. And as you increase the temperature towards before the optimum, then the frequency of effective collisions increase. These are very important points which you must write exactly in these words when you are answering the questions in the paper 2. Then in chapter 6, state the word equation and balanced equation. I've just underlined points which are sort of maybe come in the exam. I'm saying maybe. I'm not saying I can, predict anything. I'm just sort of giving you rough guidelines which I think are important topics. Then describe and explain the effect of varying light intensity CO2. And you see, the explain is the important part. The describe would probably be a graph, and describe is no biology, so you just describe the graph. But explain is the one which you need to concentrate on. Investigate the effects of light intensity, CO2, and temperature on the rate of photosynthesis using submerged aquatic plants. Then identify and explain the limiting factors of photosynthesis. You need to be looking that up. That's a very favorite topic which they like to be questioning a lot of time. Then leaf structure, large surface area, and thin, and explain how these things are adaptations for photosynthesis. Then naturally, if it's a thin leaf, then there's short diffusion distance. The CO2 enters and it immediately reaches the palisade cells where the chloroplasts are present. And large surface area, of course, increases the surface area for light absorption. So I want you to be looking into that. Then importance of nitrate ions to make amino acids and proteins, and magnesium ions to make the chlorophyll molecule. Then point 3 of the transport in plants, investigate using a suitable stain the pathway of water in a cut stem. This, of course, I need you to work on this for the paper 4. Then I like to talk about translocation, movement of sucrose and amino acids in solution in the phloem from source to sink, source where it's produced to sink where it is stored. Then identify the position of the tissues in transverse sections. Then I want you to be looking at that. If you are clear about that, I'm glad you're clear about that. Then we come on to the next chapter. The main regions of the digestive system, you must know this. You must look at a diagram and just revise that. I'm sure you all are very aware of it. Explain why most foods must be digested. Why must be digested? Because carbohydrates, fats, and proteins are the main components of food. You had roti, you had chicken kadai, you had biryani. All that contains what? Carbohydrates, fats, and proteins. And those cannot be used by the body. Yes, if you had orange juice, vitamin C will be used by the body. It doesn't need to be digested. If you had milk, it had calcium. Calcium doesn't need to be digested. So you've got to understand why most foods must be digested before they can be absorbed. Then you must know the difference between physical digestion and chemical digestion. Then you have to learn these. There are points which I want you to learn. In the mouth, what happens in the stomach. I've learned this. I said, go through this syllabus and learn this. And anything which learn I mean is you need to memorize this. So you need to memorize these points. Then you must know what is peristalsis, a wave of contraction, and the circular and the longitudinal muscles, how do they work. Then the fact that the water is absorbed from the lumen of the small intestine and the colon, but that most absorption of water happens in the small intestine. This is another very important point. Then the function of the hepatic portal vein that is taken by the liver by most of the molecules. Most of the molecules, no lipids enter the hepatic portal vein. Lipids enter the lacteals and then of course enter the blood circulation later again in the subclavian vein. So everybody must know this, be very clear on this. I want you to go through every point of the syllabus. You have 24 hours to do this. I think less than 24 hours. Then in the gas exchange, I want you to know about the internal and external intercostal muscles. I want you to know the role of the ribs, so 6, 7, and 9. And I want you to know the role of the goblet cells. Everybody not clear on anything needs to Google it and check it out. Then the uses of energy in the living organisms and the fact that in respiration we say it's the release of energy. Please do not say produce energy. Energy cannot be created or destroyed. You must be very, very clear on this. When you write in the exam, it's the release of energy. Respiration, RE respiration, RE release. Remember that. Then the word equation and the balanced equation for aerobic respiration. Then the word equation for anaerobic respiration in humans, it's a word equation. Word equation for anaerobic respiration in humans and word equation for the anaerobic respiration in yeast. Then I have said, state that the activity of the heart may be monitored by the ECG, pulse rate, and listening to sound of the valves closing. Now in 11.3, I've said name the main blood vessels. You need to look at a diagram. You must be able to name the aorta, vena cava, pulmonary artery, pulmonary vein, hepatic artery, hepatic portal vein, renal artery, and renal vein. Then I said describe and identify on diagram. So you must know this. Structure of arteries, veins, and capillaries. Thickness of the wall, arteries thicker, veins are less, they are thinner walls. Composition of the wall made up of muscle and elastic tissue. Then diameter of the lumen. Then the presence of valves in the veins. In the arteries, they're only present in the base, on the beginning of the pulmonary artery and the aorta. So this is all I want you to know on this. Then coming on to the blood chapter, I want you to learn how the platelets clot are important in clotting by converting the soluble fibrinogen to insoluble fibrin. And this, of course, forms a sort of a clot, and that prevents the entry of pathogens because in the air you have bacteria and viruses and fungus and different pathogens. So all those will be prevented from entering the body because now you've got a break in the skin. The skin is, in fact, the most protective, the largest organ of the body, and that protects you from any invading pathogens. Then describe the transfer of substance between blood and capillaries and the tissue fluid and body cells. Then diseases and immunity. This is a new chapter, new in the syllabus, and I want you to know about the malaria story. I'm not sure, maybe it could be malaria, could be something else, but just my gut feeling, I'm just saying maybe there's a question on malaria. Then describe cholera as a disease and which is transmitted in contaminated water. Why? Because cholera is the bacteria which causes cholera is passed out in the feces, not in the urine. Please remember that, because they're found in the intestine. So if those feces contaminate a water source like a well or a pond from where people take water and drink, so then of course they're going to suffer from cholera. Now in antibiotics, it says describe a drug as any substance taken into the body that modifies and affects chemical reactions. Like you take Panadol and the pain is relieved, but the cause of the pain is not relieved. Please remember. Then the use of antibiotics in the treatment of bacterial infections. Cholera is a bacterial infection. HIV or AIDS is not a bacterial infection. And antibiotics kill bacteria, but they do not affect viruses. Why? Viruses have a protein coat, they have no cytoplasm, they have a DNA or an RNA strand. Bacteria have a cell wall, they have a cell membrane, they have cytoplasm, they have enzymes in it, they have ribosomes in it, they have plasmids in it. So antibiotics would be chemical which would affect the working of any of those things, like if it destroys the cell wall, the bacteria is destroyed. If it destroys certain enzymes which are required in protein synthesis, or if it destroys the ribosomes. So anything which is going to affect the working of the bacteria or the normal processes, the respiration procedure. Please remember, bacteria respire, they do not have mitochondria, but they respire and energy is released. That's how bacteria is going to divide. And the ribosomes are where the proteins or the enzymes are going to be made. For protein synthesis, you need energy. So then this is all what we want to know about this. Then in the immunity chapter, which is also new in the syllabus, we must know the differences between active immunity and passive immunity. Active immunity is when there is antigenic exposure and then the person has measles and is exposed to the measles virus and then he develops memory cells and then he does not get measles for the rest of his life. Active immunity. Vaccination is also a type of active immunity. Passive immunity, if you get ready-made antibodies, like from mother to the fetus. Mother's lymphocytes make antibodies and then those antibodies cross the placenta and protect the fetus, that when the fetus is born and that's called a baby, then he or she is not going to get those diseases for which he has the antibodies from his mother. So that is called passive immunity. Either you get it in breastfeeding or you get it across the placenta, and in breast milk. In this chapter, I would outline these points. The function of the nephron, Bowman's capsule, glomerulus is all very new in the syllabus. This was not in there in the previous syllabus. So the role of the glomerulus is filtration from the blood of water, glucose, urea, and ions. Then the role of the nephron in reabsorption of all the glucose. You see, all of this is filtered out of the plasma. But then all this cannot be wasted in the urine, so they can't be passed out of the body as urine. So only we want to remove, the kidney only wants to remove the urea, the excess salts, and the excess water. So all this is filtered, but then it has to be reabsorbed. And this is a very active process to reabsorb all the glucose. And then the urine contains only the urea, the excess water, and the excess ions. And details of these processes are not required. Then deamination in the liver is the removal of the nitrogen containing part of amino acid resulting in the formation of urea. So urea is produced in the liver and it will enter the blood in the liver. So it will be passed out of the hepatic vein. Then it enters the general circulation, and when this blood enters the kidneys remove the urea from the blood. Remove the urea from the blood. And then it is passed out in the urine. So please remember, the urea initially was in 5 liters of blood. Now the urea is just in 50 or 100 ml of water. Urine is not that large a volume. Urine is very small volume as compared to the 5 liters of blood in which the urea was present. In this chapter, these are very new things, which are the receptor, sensory neuron, relay neuron, motor neuron, and the effector. Now, the effector can only be muscles and glands. Then you have to know reflex action, which is an automatic response to a stimulus. Then a synapse, which is junction between two neurons. Then the structure of the synapse, including the presence of vesicles, which contain a neurotransmitter. The synaptic gap, and then the receptor protein. So this is the presynaptic neuron. This is the postsynaptic neuron. The vesicles are present here, and the receptors are present here. So these vesicles will come, and then they're going to burst here, and then they're going to release this chemical, and the chemical is going to be released. The chemicals in it is going to be released, and the chemical is going to come and stick here and open the sodium channels. So the reason why this is a unidirectional is because Vesicles are only present here in the presynaptic neuron. There are no vesicles present here in the postsynaptic neuron. So that is why it is unidirectional. And that is why this is the point. Impulses travel in one direction only. So impulse stimulates the release of neurotransmitter molecules from vesicles into the synaptic gap. This is the synaptic gap. Then the molecules diffuse across the gap and bind with the receptor proteins. An impulse is stimulated in the next neuron. Explain accommodation to view near and distant objects in terms of contraction and relaxation of the ciliary muscles and the tension in the suspensory ligaments and the shape of the lens. Near, more convex. Distant, less convex. So these are the mnemonics that you need to remember when remembering this part. Then hormones, then you have to know the name. So again, this is a learn. Which organ produces which hormones? And the pancreas produces insulin, pituitary, follicle stimulating hormone and luteinizing hormone, testes, testosterone, ovaries, estrogen and progesterone, adrenal glands, adrenaline, pancreas, insulin and glucagon. Then how the following contributes to maintenance of constant internal body temperature? Sweating, shivering, contraction of the hair erector muscle, vasodilation, vasoconstriction of arterioles. Please remember, supplying the skin surface capillaries. This is an artery, divides into arterioles. And then there are capillaries. Capillaries are one cell thick wall. So things can move out, into and out of capillaries. So it is the arterioles where have muscles in their wall. And these arterioles the muscles in the wall can contract and relax. So the lumen can be altered. And when the lumen is altered, less or more blood is going to flow in it. So less or more blood is going to flow in the capillaries. So less in the skin surface capillaries. This happens when you are in a very cold room, so vasoconstriction. But you're in a hot room, vasodilation. Because the skin capillaries are a very large surface area and more heat is going to be lost from that to cool the body down. Then blood glucose control. Then why do we need to control the blood glucose? Because the blood can't go to zero. Blood glucose can't go to zero. Just like in your car, you have to have a certain volume of petrol in it, fuel in it. So glucose is your body fuel. Your cells can only respire glucose and release energy. So without glucose, you'd be dead. So we need a certain level of glucose in our body at all times. Then coming to this chapter, this is again new in the syllabus. You've got to remember all these points. Auxin is made in the shoot tip. Auxin spreads through the plant from the shoot tip. Unequally distributed in response to light and gravity. Then it stimulates cell elongation. These are points which are very basic, and these are going to be coming in the questions. Then you have to know what is gravitropism and phototropism. Then you must know what is a clinostat and how do we set that up. Advantages and disadvantages of asexual reproduction and sexual reproduction. That's a very good question which we need to revise. I'm not giving you a guest paper. I'm just giving you things that you need to revise. This is not a guest paper. Please remember that, and do not attribute this to me that her guest paper was wrong or was right or whatever. Sexual reproduction in plants, I want you to look at number 4, the flower structure pollen from insect pollinated and wind pollinated flowers. Outline the process of pollination, then the potential effects of self-pollination. In variation, there's going to be less variation in self-pollination and less capacity to respond to changes in the environments and reliance on pollinators. Then you have to know the growth of the pollen tube. That is something very factual. And that the fact that the ovules develop into seeds and the ovary develops into the fruit. So if you are having an apple, you are having a fruit. It contains seeds, and those seeds were once upon a time ovules. Menstrual cycle, release of egg and changes in the lining of the uterus. Then the early development of the zygote, leading to the formation of a ball. Then identify on diagrams the placenta and umbilical cord and their functions. And then important thing is state some viruses can cross the placenta and affect the fetus. Some viruses, not bacteria. Then we have inheritance. Importance is the discontinuous and continuous variation. And continuous variation is like height of 16-year-old boys. Discontinuation is blood group. And the important thing which you got to understand is discontinuation is usually caused by genes only, and continuous variation is caused by genes and the environment, like tall stems. Tall stems would be dependent on light and nitrates from the soil. So genetic factor will be there, but if it doesn't get enough light and nitrates from the soil, it's not going to grow tall. Then here I want you to remember 4 and 5. Sequence of bases in a gene determines the sequence of amino acids needed to make a specific protein, and the different sequence of amino acids give different shapes to protein molecules. So if this is an enzyme and then this is an enzyme, they have two different active sites. They could have the same number of amino acids, but the sequence of amino acids is different. So that is why the folding is different. That is why the shape of the active site or the depression is different in each of these two enzymes, A and B. Then I want to talk about mutation, gene mutation, random change in the base sequence of DNA. Remember, base sequence was what? ATGC. ATGC could be AT, and then GC, GC, GC. Now, if any of this changes, like this becomes, say, GC, but this is a change in the base sequence of DNA, then this is, of course, an example of sickle cell anemia in which one of this changes. Not this, it doesn't change like this, it changes in a different manner. And that results in sickle cell anemia because it is the gene for hemoglobin which changes. Gene for hemoglobin. And this, of course, results in mutations. Then again, this is called a gene mutation. Then there's also a chromosome number it can change. That is called a chromosome mutation. So gene mutation, a change in the base sequence. Chromosome mutation, a change in the chromosome number, and that is in the Down syndrome, in which instead of a normal 46 chromosomes, the person has 47 chromosomes. Mutation, meiosis, random mating, and random fertilization are sources of genetic variation. So these are the 4 points you must remember: mutation, meiosis, MMRR. Mutation, meiosis, random mating, and random fertilization. Then ionizing radiation and chemicals increase the rate of mutation. So people who do a lot of sunbathing, exposure to UV light, develop skin cancer. Then chemicals like tar in cigarette smoke, then that causes a mutation and that results in lung cancer. Points of artificial selection, you can easily write all these: selection by humans of animals or plants with desirable features, crossing these to produce next generation, selection of offspring showing the desirable features, and reputation over many generations. You can exactly repeat these words for a question. Then coming to chapter 18, why bacteria are used in biotechnology and genetic modification, no ethical concerns, and presence of plasmids. And the fact that they have a very rapid reproduction rate and their ability to make complex molecules are 4 points which we need to talk about. Rapid reproduction rate, ability to make complex molecules, no ethical concerns, and presence of plasmids. Then we have the enzymes for biological washing powders, pectinase for food production, lactose for lactose-free milk, because people are allergic to lactose. Then in genetic modification, we talk about genetic material of an organism by removing, changing, or inserting individual genes. The need for producing human insulin, then they confer resistance to herbicides and confer resistance to insect pests and provide additional vitamins. Then the advantages of genetic modification, limited to modifying crop plants and bacteria. Last chapter I want to talk about is you have to know the flow of energy through food chains. This is a very favorite question. Construct simple food chains and construct and implement pyramids of numbers, biomass, and energy. The nutrient cycles, we have our favorite, the carbon cycle and the nitrogen cycle. You must know these points. You must go through these points. Please read through these points and remember these points. Then ecosystems and biodiversity. There are not many things that I would like to. I'm not sort of zoning into that much. So describe a population, describe a community, describe an ecosystem, describe biodiversity. So you must know these definitions. Water pollution, you must know this, all the reasons for water pollution and how it causes eutrophication And all these points, please go through these points and learn them, preferably. Just quick recall them. In conservation, I would like you to remember the forest story, education, protected areas, quotas, and replanting. And in the fish stocks, I want you to know education, closed seasons, protected areas, controlled net types, and mesh size, quotas, and monitoring. Then you must know the definition. What is a sustainable resource? Which is produced as rapidly as it is removed from the environment and so that it does not run out. Please remember this is an update by the Hodder Education. You can just Google it. These are the topics which are no longer in the syllabus. You keep on asking me these questions, and I'm surprised as you are not aware of this. World food supplies, malnutrition, causes of tooth decay, kidney dialysis, structure of the brain, drugs that effects of heroin and heroin, then syphilis, advantage of breast milk, bottled milk, birth control, parasitism, support movement, and locomotion is all being removed from the syllabus. You waste a lot of time on doing stuff which is not in the syllabus. Thank you and best of luck, and wish you all the very best in paper 2 and paper 4, which is also very nearby. Thank you very much.